Good evening, everyone. It is a pleasure for me to be here tonight to introduce Jocelyne Bloch and Grégoire Courtine, two stars in neurotechnology. In fact, I don't think they need to be introduced, knowing the number of publications they have had in the magazine Nature and Science. We created the DefiTech Foundation 20 years ago with my husband to contribute to the research and development of products and technologies to help people with disabilities. Thanks to Patrick Ebicher, 10 years ago, we had the chance to meet Grégoire and Jocelyne at EPFL. We have been impressed immediately by their dream to have paralyzed people walk again. Since then, starting with rats, they are now able to have paralyzed people walk again. So, enough said. I let Grégoire and Jocelyne speak about themselves.
Merci.
Thank you, Sylviane.
Thank you, Sylviane, for this nice introduction. So, good evening. Last May, a lot of you have certainly heard about this paralyzed man with a spinal cord injury who was able to walk thanks to a digital bridge linking his brain with his spinal cord. So today, we are going to tell you the story of how it happened. So first, I would like to tell you about the spinal cord injury, just a few words. So when you have a spinal cord injury, your brain sends a command to your legs, but it's interrupted at the level of the spinal cord injury. So many years ago, already, Grégoire went to the US and he started studying the idea of reactivating the low part of the spinal cord in order to activate the legs.
When she said a long time ago, it's 20 years ago, it was very rudimentary at the time. Imagine the rat completely paralyzed, and at the time we were just applying wide field stimulation of the spinal cord to restore walking. Worked very well. Steam on, he walks, turn off, the animal can't step, back on, immediately you resume walking. So then I had the dream of the scientists who would take this to human. But of course there is a huge step. So for me the first important step before going to human was to understand the underlying mechanism. So we spent quite some time modeling all the structure of the spinal cord, the electrical field elicited by the stimulation, to find out eventually that the electricity does not penetrate the spinal cord. It flows on the side, as you can see, where it will stimulate what is called the sensory afferents, specifically coming from muscle, proprioceptive afferents, because they are the less resistant to current. So we understood that this stimulation that looks very unspecific is actually very specific for a very specific pathway, which allows us to operate a transition from this empirical stimulation to a mechanism-driven stimulation. What do I mean by this? Since we activate the sensory fiber that enter through the dorsal root of the spinal cord, we could target them individually with a spatio-temporal stimulation pattern and reproduce the way the brain naturally activates the spinal cord in order to walk. So what we call a biomimetic stimulation of the spinal cord. This is a Princeps experiment. The rats would be paralyzed. We apply continuous stimulation as I showed you. The animal step. But it's 1 week after the injury. Spinal cord is very weak. The animal collapses. We turn on the spatio-temporal stimulation, and immediately you restore walking and increase even the vigor of the movement. That was the turning point for us. We understood how to dialogue with the spinal cord to restore walking with mechanistic understanding. We published this data. It was propelled throughout the world. Imagine the little rat that ended up in the front page of the New York Times, which at the time for the 70s was a huge pressure, actually. Rapidly, the pride vanished, and then the pressure to transit from the sophisticated laboratory to a therapy that can help people came. That's when I realized that there are two important challenges. First, we needed to develop the technology for human, and second, we needed a clinical partner. First, I happened to meet someone you may know, in June 2011, Patrick Abisher welcomed me in his office and told me that the EPFL had developed a center for neuroprosthetic, so a center really dedicated to developing neurotechnology. So it took me a few months to bring all the laboratory at EPFL. And he told me also, don't worry, I also have formed a wonderful neurosurgeon, and she will be able to help you to operate the path from rats to humans.
That was 2012.
Correct.
So the first time I met Grégoire, he was in his office. I saw the rats walking, very enthusiastic. He told me, can we go tomorrow to the human? And I was also very enthusiastic, but you have to imagine that to apply this special stimulation, what we call biomimetic, so to mimic the natural way the people are walking, it was impossible with the implants we had available. So we had to think about it and to partner with Medtronic to repurpose implants that they already had. So we used paddle leads, electrodes that are normally used to treat chronic pain, and we used a stimulator that is normally used to treat people with Parkinson's disease. And together, with many engineers and a complicated chain of commands, for the first time, we had a device that was able to do what we call closed loop. It means that we can take into account what the person wants to do and stimulate at the right time, at the right spot, in order to make him walk. 2016, so 4 years later, we were ready. That was the first surgery on David, the first man you saw in the first picture. We were all very scared. Will it work the same as it works in rats? And it immediately worked. So you see here David with the stimulation, who is immediately able to step. If you stop stimulation, he stops walking. And if you resume stimulation, he'll walk again. And once we thought, why do we make it so complicated? Do we need such a complicated biomimetic stimulation, or would we also be able to apply current all over the spinal cord, and would it work the same? So we knew, testing both stimulation patterns, that this biomimetic stimulation was the way to go. You see underneath a continuous stimulation, David cannot walk properly. With this complicated but very efficient way to stimulate, he was able to walk.
But tell me, Jocelyne,
yes,
what did we learn during this first step in humans?
So we learned that it was possible, that was the first path to go, the way to go, but that it was too complicated. So as I told you, the implants were not dedicated for that, and all this long chain of command in order to be able to give this appropriate stimulation was not going to be sustainable.
And you probably all know the complicated relationship with industry, especially big companies like Medtronic. So very rapidly, we understood with Jocelyne that in order to develop a treatment, we had to control our destiny. So 2014, we launched a startup, at the beginning with a lot of naivety, and eventually it became Onward Medical, a company that is now in the stock market. And the goal of Onward Medical was to build a purpose-built neurostimulation platform for precise neurostimulation of the spinal cord. So this stimulator linked to a lead, you have a programmer outside that is basically a computer that can communicate wirelessly with the implantable pacemaker. This allows the patient to control the stimulator with a watch and have this real-time control over the stimulation of the spinal cord for the very first time with a very precise system.
Mm-hmm. So everything became simpler and also much more personalized. So for each patient, you have the dedicated electrode, you put it right on the right place, and you have very good results immediately. So you see here the spine of each individual, and you can link it with the stimulator.
So that's the surgery. Of course, as Jocelyne said, it took like 3 PhD students from EPFL to configure the stimulation, so not a scalable therapy. Hence comes the challenge. How do you develop basically a stimulation strategy that everybody can use? So what we did is use AI. So here you see, I don't know why the video doesn't work here. We have some problem with the video, I think. We're missing a lot of things. So what you should see here, I don't know where it's stuck, it works here, but not up here. So you should see that automatically you have an AI applying the stimulation and searching where the stimulation should be delivered in order to elicit the movement. And then you do not see the video, so there are no video working. So here it's what is called a patient programmer, so it doesn't work. Do you want to relaunch the PowerPoint, maybe?
I think you should have a little drink.
Enjoy your drink, we're going to make a little pause.
Because it's a pity if everything is based on video, so if you don't see them, I mean, we... Au début, ça marchait?
Je crois, oui.
Je regardais qu'ici.
Ah, voilà.
Ah, yeah.
So here the AI is searching how to stimulate to induce isolated movement, as you can see, knee extension, flexion, etc. So we are calling that individual block, which we inject in this time scheduler, so we can say when you want an extension or a flexion. And then it's controlled by an handheld tablet. So the physical therapist can personalize the stimulation to help the patient. For example, more hip flexion, increase the amplitude very easily to have a better flexion. And this system can be used outside the laboratory environment. So the patient can use a watch, as I told you, to trigger the stimulation. The program that will help to do not only walking, but other activities. And because you understood that we must synchronize the stimulation with the intention, we use devices, this one, absolutely not sophisticated, to capture the intention, the clicker. I click, I do a swing phase. Or a bit better, we can measure the residual movement with an inertial measurement unit that says, okay, you kind of want to do a swing phase, I will stimulate to do a swing phase. And using this technology, the participants are able to step outside.
Stim on.
Okay, start message sent to implant.
So as you can see, it's not that he's walking normally, but for the first time, an individual with paralysis is able to take voluntary steps independently outside the laboratory environment.
So he's not only able to walk, but he's also able to perform other things like swimming, for example. There are different programs. They can bike with also stimulation. And they do a lot of different activities in order to be able to be better. So what was published with these first pioneers was then a lot in the medias. You've probably heard this in 2018. The BBC, all the medias in the world spoke about it. And finally, it helped us a lot, because thanks to this media and this press coverage, we, again, Patrick Abisher, introduced us to Sylviane Borel and Bobo, he's the husband of Sylviane. And together, we could found a center dedicated to the research we were performing that we called NeuroRestore, and it really helped us to be better.
And for you, some of you are politicians. Normally, laboratory is very simple. You have a boss, and you push research. Here it's different. We are two co-directors with very different expertise. I'm more like a neuroscientist engineer. She's a neurosurgeon. And when we created NeuroRestore, we really wanted to leverage this synergy. So we capture our expertise in the philosophy of the center. In all the studies we perform now at NeuroRestore, we go all the way from understanding the mechanism of action, a lot of work in mice, rats, scaling them up in non-human primates, eventually proof of concept. And because there is onward, we can really scale up the therapy when we see that they're effective in humans. And this development model has been incredibly effective and really enabled by this unique center that we have been able to create.
So thanks to it, we could go one step further and make people walk even better. So you remember, Grégoire told you that to walk, they need either clickers in order to have one leg after the other, or these inertial measurement units that they have on the shoes. But this still is a bit frustrating because it's not that natural. There is always something in between that needs to be activated in order to make the patient walk. So that's when we had this idea of having something much more natural that we would call the digital bridge. So what is it, G?
So this started actually with experiment in non-human primates because we needed more advanced mammal models. Here, Jocelyn inserted electrode in the region of the motor cortex that encode leg movement. So we had a wireless recording for the very first time, sent to an external computer that runs an AI that decodes the intention to move and use the exact same neurostimulation platform that we have been using in human to elicit the desired movement. So a digital bridge that turned the thought into action. And normally you say, well, that sounds crazy. Decode the intention. So I'm going to do a little exercise for you. This is the type of recordings you can obtain when the animal is standing and then walking. So you have to be the algorithm. You're going to use like some statics. This is the activity of cells in the brain. And you have to decode when the animal goes from standing to walking. So you can hear this beautiful symphony that's called walking. And this is why we can decode very robustly the intention to move the leg because they're so well encoded in the motor cortex. So we use this approach. This is a model of transitory paralysis of the right leg. You see the right leg cannot be moved. There is 2 minutes of calibration only of the decoder in this case because it's very simple. Does the animal want to do a stance phase or a swing phase and how much? And you see the animal now just thinking about it, stimulate itself, I can step, we turn off the digital bridge and it becomes paralyzed again. And in my experience of a scientist, this was the more emotional moment of my career. It was so complicated to make this work technologically. But see a paralyzed monkey walking, my dream was to see this in humans. Yet, I'm working with a neurosurgeon who does not like to insert electrode in the human brain.
In the public, there is one who is inserting brain. So I won't criticize too much, but I thought that someone with a who is paralyzed because of a spinal cord injury, but with an intact brain, should at least at this stage not receive an implant that is penetrating his motor cortex. So we decided to work with an implant that could be just put above the motor cortex in order to record the activity and then to transmit it to the spinal implants. And we teamed up with a team from France, Grenoble, they developed this device that is called Wimagine, and that is exactly doing what we wanted. And the good thing also, it's a passive device, everything is closed, no battery to change. When it's put in, you just close the skin and then it's done.
What was the next step?
We had to find a patient because, you know, we have to find the number one willing to try this new technology. So we met Gert-Jan, a Dutch young man who had this bike accident a few years ago with a cervical injury. And when we met him, that was the way he was able to walk. So there was very little movement.
So Jocelyn implanted the digital bridge. So you understand this is this 2x64 electrode that replaces the skull. So with imaging, we localize the region of the motor cortex involved in leg movement. So you imagine leg movement, we measure this, she positions the implant, it is sent to an external computer, but fully wirelessly, that runs the AI, that decodes the intention to move, and the AI translates the decoded thought into stimulation pattern to activate the spinal cord and then elicit the desired movement. So it's all fully wireless, fully implanted. And this is the very first moment Gert-Jan, our first test pilot, is trying to do a hip flexion of the right side. So you see the technology on the right. You're going to see the decoder. You see the little triangle. It goes from blue, rest, to bottom right, which is a hip flexion. So the more you think about doing a hip flexion, the more it will stimulate the region of the spinal cord that want to elicit a hip flexion. So you see two movements. This is the first one. Second one, you will see the dots going bottom right, and it translates into a proportional stimulation of the region of the spinal cord to elicit hip flexion. So it works right away. It was really remarkable for us to see this. But the best is to let Gert-Jan tell you how he can use this stimulation, this digital bridge, in order to have very natural control of his paralyzed legs.
I have a spinal cord injury at C5, C6, and recently I've been implanted by a brain-spine interface. So here's an example. I can start the stim with my brain. I can talk while doing it, and if I want, I can also maintain the stimulation while talking. I'm maintaining now. So this feels more natural. I can control the step length. I can stop here. I can make it bigger. It's freedom, which I didn't have with the foot sensors before. I can stop right now, and I can continue whenever I want.
So this was a journey to the digital bridge, but with Jocelyn we thought, well, can we do something else?
Yes, if you can walk with a digital bridge, why couldn't you move also your arm with a digital bridge? And you know, a lot of people with a cervical lesion cannot move their hands, they are tetraplegic, and any movement that they regain would also be very helpful for their daily activity. So we show you in exclusivity, it's the very beginning, but we applied exactly the same thing for the upper extremity. The principle is the same, so the same type of system that is implanted in the brain, above the brain, and also electrodes that are this time not located in the area of the spinal cord that is controlling leg movement, but this time is for hand movement. So you see here this patient who will get everything to activate his left arm. So here again, it's the very beginning. We are starting rehab with him, but for the first time, after 2 years of complete paralysis of the left arm, he's able, just by thinking about it, to open his hand.
That was pretty cool to see it, I can tell. Day one.
So that's a little bit of what we are doing, not both of us, but the whole team, and also thanks to Sylviane, her husband, and all the people who helps us. A lot of engineers, a lot of people in the medical background can together work in order to make it work. But, I mean, still, we are working a lot, but it's not enough to treat the whole world. Huh?
Now, this is like a really unique innovation platform. So we are super happy about this. And we believe that for translational medicine, having not collaboration, but truly under the same roof, engineer, neuroscientist, and medical doctor of various expertise working together is unique and key. So if you want to achieve translation, especially in neuroscience, you have to promote this type of model. But as Jocelyn said, it's frustrating because every day we receive maybe 10 to 20 requests, and more because we are more and more in the media, and it's not yet available. We're talking about 1 patient, 2 patients, proof of concept. And this is why we founded Onward in 2014. And today, Onward is about to launch the first pivotal trial in the US and Europe that is called Empower Mobility, in order to turn this technology into a widely available technology for all the patients who need it. And we really hope we will succeed in this translation. Thank you.